Hello everyone, in this video we are going to discuss about pulmonary surfactant. We know pneumocytes line the alveoli. These are of two types, type 1 and type 2. Type 1 pneumocytes are about 97% of all the pneumocytes. They line the alveoli and they are squamous type and they are very thin. So they help in gas diffusion and exchange. Coming to the type 2, they are very less in number but they are cuboidal and clustered. Though they are very less, they help in secretion of the pulmonary surfactant, which is the main important function of the type 2 pneumocytes. They also help in regenerative function and repair function. They produce type 2 and type 1 pneumocytes, the pulmonary surfactant decreases the alveolar surface tension, so they inhibit the atelectasis and inhibit end respiratory collapse and increase the compliance, that is expandability of the alveoli. So these the type 2 pneumocytes are cuboidal and type 1 are the squamous in type. So in addition to this, there are other cells called as club cells. These are non-ciliated low columnar to cuboidal cells with secretory granules. The secret club cell secretory protein, and they help in detoxification and also in the repair. So the three types of cells are type 1 and type 2 pneumocytes and the club cells. So coming to the surfactant. So the main surfactant is the pulmonary surfactant is mainly made up of lipids, 90% and 10% of proteins. The key lipid is the dipalmitoyl phosphatidylcholine, also called as DPPC. It reduces the alveolar surface tension, and in addition to the DPPC, it also has SPB and C and SPA and D. SPB and C are the hydrophobic proteins and SPA and D are the hydrophilic proteins. B and C help in stabilizing the surfactant, A and D help in immune defense. So this is the hydrophilic part. Outer part is the hydrophilic part, inner part is the tail is the hydrophobic part. So here we know, so in the children during the gestation age, from 24 weeks of gestation, the pulmonary surfactant will start produced. By the 35 weeks, it will be completely matured. So it will have two parts, lecithin and sphingomyelin. Initially, by 24 weeks, it will be in the ratio of 1:1. As the gestational age progresses, at 35 weeks, it is almost about 2:1, lecithin to sphingomyelin ratio. It indicates lung maturity. Why this is important? Because in cases of premature rupture of membranes or any premature delivery, when we take amniotic fluid thing and we will do this lecithin to sphingomyelin ratio, if it is 2:1, we don't need to induce any corticosteroids. If it is less, we have to give corticosteroids in case of premature rupture of membranes or premature delivery, because in those if the ratio is less than 2:1, it indicates lung is still immature. So we will see with and without surfactant. Without surfactant, what happens is the alveoli tends to collapse, and with surfactant the alveoli tends to expand, because surfactant will help in reducing the surface tension. So we know, according to the Laplace law, pressure is equal to 2 into surface tension by radius. So if the radius is very less, the pressure will be very high, so they tend to collapse. But with the surfactant, this is the green thing is the layer of the fluid on the alveoli. The surfactant will disrupt the hydrogen bond between the water molecules and helps in expansion and helps in increasing the compliance. So this diagram shows the same. So usually if the radius is small, it should collapse because of increasing the pressure, but it doesn't collapse because surfactant per unit area is more in this, so they doesn't collapse and inflate at a faster rate compared to the larger alveoli with less amount of surfactant per unit area. So this is the pulmonary surfactant. So main thing is lecithin to sphingomyelin ratio 2:1 indicates the lung maturity, and at 35 weeks lungs become mature. So type 2 pneumocytes produce the pulmonary surfactant.